Hello, welcome friend. Welcome to our channel Quality Guru. Friend, today our topic is best practices of microbiology in pharmaceutical industry. Best practices in pharmaceutical microbiology involve establishing stringent aseptic conditions through proper laboratory design, personal and material hygiene, and strict adherence to standard operating procedure, that is SOPs. Key practices include validated environmental monitoring, meticulous sample handling and analysis, comprehensive personal trainings, meticulous data integrity, and proper use and storage of reference cultures and testing materials like biological indicators. Best practices in pharmaceutical microbiology are significant because they ensure patient safety by preventing contamination and verifying product quality, maintain product efficacy, guarantee compliance and stringent regulatory standards, and protect a company reputation and finance by preventing recalls and outbreaks. Laboratory design and environment, separation and containment. The microbiology laboratory should be separate, dedicated space away from production area to prevent cross contamination and mix up. Space and access control. Adequate space is needed for handling sample, media and organism with restricted access only for authorized personnel. Environmental control. Maintain proper temperature, humidity and clean air. Say for example HEPA, high efficiency particulate air filter with separate air handling for critical areas, especially those used for sterility testing. Easy disinfection. The laboratory design should facilitate easy and effective cleaning and disinfection to minimize contamination risk. Personal and hygiene, gowning and production. Strict adherence to gowning procedures, including sterile garments, gloves, masks, and goggles, is essential for personal working in controlled areas. Hand hygiene. Thorough hand washing and sanitization with filtered alcohol solution should be performed before and after activity. Training. Personnel require regular training in aseptic techniques, data integrity, contamination control, and specific SOPs. Health. Employees with illness that could pose a risk should report them before entering into the laboratory. Aseptic techniques and operations. Laminar airflow hoods. All microbiological analysis must be performed under aseptic condition, typically within a laminar airflow, that is LAF hood. Disinfection. Before and after use, surfaces and equipment including gloves and other material must be disinfected to prevent contamination. Control movement. Avoid unnecessary talking, movement or activity within sterile zone to minimize airborne contamination. Method and documentation. Validated methods. All microbiological method, that is sterility testing, microbial limit tests, endotoxin testing, bioburden testing must be scientifically validated for accuracy and reproducibility. Standardized procedure. Clear, detailed SOPs for all processes including media preparation, equipment cleaning and waste disposal are critical. Data integrity. Maintain robust data integrity practices, ensuring that all test results and observations are accurate, complete and reliable. Risk assessment. Conduct microbial risk assessment for operational clean rooms to proactively identify and control contamination sources. Sampling, storage and materials. Sample integrity. Sample must be immediately labeled, handled carefully to prevent contamination and analyzed as soon as possible or stored appropriately. Culture management. Maintain a well managed culture collection with subculturing performed regularly and access restricted to authorized personnel only. Biological indicators. Biological indicator must be treated as raw material with strict quality control, including testing for count and identity, and control use and storage according to SOPs. Do's in the microbiology lab. Wear appropriate PPEs. Always wear a lab coat, glove, head cap, and face masks when handling microbial activity. Maintain aseptic condition. Work with proper gowning and use laminar flow hood for microbial analysis. Disinfectant and sterilize. Disinfectant work surface before and after work, and sterilize tools like inoculating loops. Use proper waste disposal. Place contaminated material in a designated biohazard waste container. Follow standard operating procedures. Adhere to detailed, validated, and standardized method for media preparation, cleaning, and testing. Handle sample correctly. Label sample immediately, keep them safe, and analyze them as soon as possible. Work efficiently. Allocate sufficient time and understand steps to avoid mistake. Be prepared. Keep all necessary equipment and supplies in good order. Wear appropriate clothing. Wear closed toed shoes and long pants to protect yourself from spills. Don'ts in microbiology lab. Do not work when ill. Avoid entering into the lab with cuts, wounds, or any illness. Do not eat, drink, or smoke. Prohibit eating, drinking, smoking, or chewing in the testing area. Do not wear street clothes. Do not wear dirty aprons, uniforms, or any other unclean articles into the laboratory. Do not bring food into the laboratory. Keep all food items and personal articles like jewelry and watches out of the lab area. Do not use unsterilized equipment. Never use contaminated or unsterilized media, reagent, water, or equipment. Do not handle culture with bare hands. Always work with gloves and appropriate tools. Do not make error. Avoid rushing and don't dismiss expected result. Instead, investigate their root cause and define a concrete kappa. Do not work alone in high risk experiments. Ensure someone is available to assist in case of an emergency. Do not bring unnecessary items. Keep personal items and unclean articles out of the laboratory. Significant outbreak of the best practices. Patient safety. The primary significance is to protect consumer from infections and adhere health effect caused by harmful microorganisms. Product quality and efficacy. Best practices ensure that drugs meet high standard for purity and effectiveness, preventing microbial growth that could compromise the product. Regulatory compliance. Adhere to validated procedures and standards is vital for gaining approval from regulatory bodies like the FDA and the EMA. Brand and financial health. Preventing contamination incidents, avoid costly product recall, legal issues, and reputational damage. Process control and validation. Rigorous testing and monitoring are cornerstones of quality control, ensuring that manufacturing processes are effective and that product meets safety standard. Reliable and accurate data. Best practices ensure that tests are performed correctly, generating reliable data for decision making and supporting product release. Aseptic environment. These practices support aseptic processing and sterilization technique, minimizing microbial presence in the manufacturing environment. Public confidence. By consistently producing safe and effective pharmaceuticals, company build and maintain public trust in their products. Key takeaway. Good microbiological practices encompass safety rules and procedure to prevent contamination and infection in microbiology lab, including wearing appropriate PPEs, using aseptic technique, performing proper hand hygiene, sterilizing equipment and workplace, avoiding consuming food or drink in the laboratory, maintaining a clean and organized environment, and following safe disposal method for biological waste. Reference guideline: Microbiological West Laboratory Practices USP Chapter 1117 Value and recent changes to a guidance of quality laboratory practices. Thank you, thanks a lot, and keep watching our channel Quality Guru. It's a small initiative and knowledge sharing platform for our pharmaceutical professional. Like, comments, and subscribe please, more and more. Thank you.